Hi, in this video we're going to talk about meiosis. Meiosis is the creation of gametes or sex cells, and it's spelled M-E-I-O-S-I-S. You should have a piece of paper that, let me back up a second. In this video we're going to talk about meiosis. Meiosis is the process of creating gametes, which are sex cells, egg and sperm. So you're going to fold your paper in half 3 times without unfolding it. You're going to fold it in half, hamburger. And when you open it up, after folding it in half 3 times, you're going to have 4 squares or 4 locations. We're going to draw a prophase 1 in this location. After prophase is metaphase 1. And after metaphase is anaphase. Good. 1. After anaphase is telophase 1. Now if you turn your paper sideways, right along the edge here, we are going to write cytokinesis 1. All right, and that happens after telophase. After cytokinesis 1, we have prophase 2, metaphase 2, and anaphase 2. Telophase 2. And last but not least, we have cytokinesis 2. All right, so let's start drawing these. In prophase, we have a cell, and that cell has a nuclear membrane that is dissolving. You should write that down. Centrioles that are moving to the poles. You should write that down. Spindle fibers that are forming. You should write that down. But the most important thing that's going on here is we have chromosomes, a large red, a medium red, and a small red. The small red is not coiled up yet. We have a large green, a medium green, and a small green that's not coiled up yet. So in prophase 1, we have chromatin that coils up into homologous pairs. This is a homologous pair. It's 2 chromosomes that code for the same genetic information. It's also called a tetrad. So this is a homologous pair. That's an A. Also known as tetrad. The reason why it's also known as a tetrad is you can see there are 4 sister chromatids in this homologous pair. Tetra stands for 4. That's why this is called a tetrad. In meiosis, we've got our cell membrane. We have our centrioles that have made it to the poles. Now this is where things become a little bit different. We have our large and our medium and our small green chromosome that line up, but they don't line up single file like they did in mitosis. They actually line up in homologous pairs, but they don't line up with all the red ones on one side, all the green ones on the other side. They line up on either side, but they do line up in homologous pairs. So please write that down. You also need to note that, there we go, that the spindle fibers connect the centromere to the centriole, but they only attach it to one centriole on the side of that chromosome. Okay. So in metaphase, two things are going on. Chromosomes line up in homologous pairs. Spindle fibers connect the centriole of a homolog. So one of the pair of chromosomes, one of these is called a homolog. One of the replicated chromosomes is then connected to the centriole by its centromere. In anaphase, the cell begins to elongate. The centrioles have remained at the poles, and you can see here that the large green chromosome is going to go that direction. The small green chromosome is going to go in that direction, and the medium green chromosome is going to go in that direction. It's actually pulled by the centromere. That's why you see that funny looking shape that you do. So my red, since my large green went this way, my large red is going to go the other direction. My small red is going to go in this direction since the other green one went that way. And my medium red is going to go this way. These are being pulled by spindle fibers, exactly. So the spindle fibers are going to pull these chromosomes toward the poles. They're going to pull them towards the centrioles by the centromeres. So in anaphase, the one thing that you should write down is homologs, this word over here. Homologs are separated in anaphase 1. So in telophase, we're going to draw that peanut-shaped cell, something like this. Our nuclear membranes are beginning to form. The centrioles are at the poles. The spindle fibers are beginning to dissolve. So you should write those three things, or those two things down. The nuclear membranes beginning to form. The spindle fibers are dissolving. Now, on this side we have the large and medium red. So we have the large and the medium red chromosomes. And on this side we have the small red chromosomes. So let's make this medium a little bigger. There we go. And on the other side we have the green. So we have the large green on this side because the large green and the small red were together. And we have the medium green on this side. And we have the small green over here. And these are in these locations because of the way they were arranged in anaphase. Now, once telophase is complete, find my cursor here. Can't find my cursor. There we go. Okay. So once telophase is complete, cytokinesis takes place, and it cuts this one cell into two. So cytokinesis 1 divides the cytoplasm of the cell. Now we're going to continue on. We're going to use one of these cells in prophase 2. We actually have both of these cells going through what we're going to describe here, but we're going to draw just one of those cells going through this process. So this nuclear membrane begins to dissolve. The centrioles move to the poles. Spindle fibers begin to form. You should write those three things down. Centrioles move to the poles. Spindle fibers begin to form, and the nuclear membrane breaks down. The last part, but the most important part of this, is we have replicated chromosomes that are already coiled up. So that you should write down as well. the replicated chromosomes are already coiled up in prophase 1. In prophase 1. So moving on to metaphase 1, our centrioles have made it to the poles. Our chromosomes are going to line up along the center of the cell in no particular order, but they're going to line up single file. And when they line up single file, the centromere is going to be attached to the centriole by a spindle fiber on each side. So in metaphase, you need to write down two things. The chromosomes line up single file, and spindle fibers connect the centrioles to both connect the centrioles to the centromere on both sides of the chromosome. So talking about anaphase 1, just like prophase 1 and metaphase 1, look very much like anaphase that's going on in mitosis. We just have half the genetic information. So the centrioles are at the poles. And we actually separate the sister chromatids into single chromatids. That is incorrect. We separate the sister chromatids into single chromosomes. That is correct. So we've got our small one going that way and small one going this way. And I have the wrong color. Let's fix that. Let's get green. There we go. So we have our green, small, going both directions. And these are being pulled by the spindle fibers, again, that are connecting the centromere to the centrioles, just like so. The last phase in meiosis is telophase 2. And telophase 2, again, we have a peanut-shaped cell. We have a nuclear membrane forming on each side of the cell. We have centrioles that are there. We have spindle fibers that are dissolving. So you should write those things down. The nuclear membranes are forming, and the spindle fibers are dissolving. We also have stuff going on with the genetic information. So our large single chromosomes are uncoiling into chromatin, and our medium single chromosome is also uncoiling into chromatin, as well as our green. So again, the single chromosome, each single chromosome is unwinding or unraveling into chromatin. Now, what happens after telophase 2 is cytokinesis 2. After cytokinesis 2 happens, we now, after this entire process happens, we have 4 genetically unique daughter cells. These are called gametes. Good. All right, that is meiosis. Let's talk a little bit about some terms. So let me let's go to a fresh page. So we have these terms. We have haploid. Haploid is haploid cell. A haploid cell is represented by N. This is a cell containing one set of chromosomes. And these are gametes. Gametes are egg and sperm. There's also cells called diploid cells. So a haploid cell and a diploid cell do not have to be capitalized. I just started doing that when I wrote haploid, so I'm going to continue. So a diploid cell is a 2N cell. And the 2N means that this is a cell containing 2 sets of chromosomes. And these are body cells. So this is every cell in your entire body except for your sex cells, except for your egg if you're female or sperm if you're male. So your liver cells, your skin cells, your blood cells, all of these are diploid cells. In humans, there are 46 chromosomes in a diploid cell. So in all of your body cells, there are 46 chromosomes. So how many chromosomes in humans in a diploid cell? So, excuse me, in a haploid cell? You're right, there are 23. So when your body and my body are making eggs and sperm, we are reducing the number of chromosomes to half. So these are in gametes. So meiosis creates gametes. And that should be all you need for this particular topic. So if you have any questions, let me know.